of the Protein Society. I'd like to welcome you all to our webinar on liquid-liquid phase separation of proteins and their role in pathology, which has been organized by Fabrizio Chiti. And again, I'll just tell you a few things about the Protein Society while people are still joining us. So the Protein Society is an international academic society. We have members in approximately 30 countries. We publish the journal Protein Science, to which I hope you'll all consider submitting your work. And our major activity is to run an annual symposium each summer. And so last summer, we were scheduled to do this in Sapporo, Japan. We were going to run the World Conference on Protein Science, but obviously that meeting, like many other scientific meetings, was canceled due to the pandemic. And as we work on planning our next major event, we've been looking for ways to sustain our community of members and also share exciting advances in science. So we've been doing that with our webinar series, which is a new initiative, and that's going extremely well. So we've now run a number of webinars. The first three are listed here, and these included fantastic scientific talks from this list of speakers. And then our fourth webinar, which we held just last month, was particularly special to me. This was organized by Bill Clemens, who you can see here, and by the Protein Society DEI committee. And this webinar featured the panel of Protein Society stars, who you can see here, and they spoke about issues of diversity, equity, and inclusion in STEM, in our field, and in their own lives. And their talks are recorded and available on our website, as are many of the other webinars on scientific topics. And I encourage you to listen. These experts discuss challenges that we face in making protein science more welcoming for everyone, and ideas and initiatives that they describe for making progress in this area. In addition to their talks, the speakers at the Diversity, Equity, and Inclusion webinar provided a variety of resources that are now posted on our website as well, and that you can see here. One more note on that, I encourage you all to visit our diversifiedproteinscience.org site. This includes a database of protein scientists from underrepresented groups. And if you are such a person, as I am, as a woman in protein science, please consider going to this site and listing yourself. This will allow people who are organizing conferences and constituting committees to find you and access your talents and expertise. And if you are an event organizer, I'd encourage you to check here for excellent people to include in your event. Okay, so our next webinar will be February 3rd. This is being organized by Chris Ball from the Institute for Protein Innovation, and will be about new capabilities in computational protein design. That's a topic close to my own heart. You can see the terrific lineup of speakers here, and registration for that is available online. And it is not too late to organize your own webinar. So many of the events that I've been telling you about were organized by Protein Society members. And if you go to our website, following information that we'll put into the chat, you can read about the guidelines for submitting your own proposal, where you provide the speakers and the science, and we provide all of the supporting logistics and advertising. Okay, so next July, we will definitely run our annual symposium. We're working out what the format of that will be now, so stay tuned for more information. But regardless of the particular format of the meeting, we will certainly be offering outstanding scientific content and career development opportunities for young scientists, and I really hope many of you will attend and participate. Okay, so with that, I'm going to hand things over to Protein Society member and webinar organizer, Fabrizio Chiti of the University of Florence, and he will explain our format and our protocols and introduce the first speaker. Over to you, Fabrizio. I will stop sharing.
It's a pleasure to be here, and it's a pleasure to have a webinar on this topic, liquid phase separation, because when I applied to organize this webinar, I thought that the Protein Society could be a good scenario for presenting this phenomenon, because it's something that occurs in vitro in the test tube when a protein is purified from the cellular context, but it's also present in vivo because many membraneless organelles are actually involved liquid-liquid phase separation. And actually, just to introduce the topic, let me share my screen so that, okay. And okay, so just to introduce the topic, what is liquid-liquid phase separation? So we have known for a long time that proteins can aggregate, and protein aggregation is associated with a number of diseases. And in conventional protein aggregation, we start from a monomer, which can be a folded protein, a partially folded state, a natively unfolded state like an intrinsically disordered protein. And at the beginning, we have some disordered aggregates, which then convert into something with a more structured beta sheet content. This is what we call the nucleus, which is the shortest fiber able to nucleate the growth of a fiber. And then by addition of monomers or aggregates, we can have the growth of this nucleus into a well-defined fiber. This is conventional protein aggregation, and the mechanism that I'm showing in this slide is the nucleated conformational conversion, which is one of the many mechanisms that have been described in protein aggregation. But conventional protein aggregation is maybe only part of the story because we have liquid-liquid phase separation. So proteins can assemble into something that is not solid, and that is not in a solid phase, but in a liquid phase. And the phenomenon is called liquid-liquid phase separation because actually these proteins can form droplets in a liquid phase, which is well separated from the solvent, from the surrounding solvent or cellular milieu, if you prefer. And so we have two liquid phases here, the assembly of the protein, but also the surrounding milieu, which is also liquid, of course. And this is why the process is called liquid-liquid phase separation. And in this droplet, the proteins maintain their liquid phase, but at some point, it can be that a nucleus forms within this droplet, and eventually the fibers can grow from these droplets to form fibrils. And it's a major question whether protein aggregation occurs with the first or the second mechanism. Maybe there could be some crosstalk between the two processes, so we might have that both are at work, or it might be that one is prevalent. And that depends on the condition, depends on the disease, depends on the protein, and it's a major question to answer in the field, of course. And this process, as I said before, occurs both in vitro and in vivo. So if you take a purified protein, you might observe a liquid-liquid phase separation. This has been observed for tau, it has been observed for TDP43, it has been observed for FUS and for many other proteins. So it's a process that can occur for purified proteins, but also in vivo, there is some physiological relevance because there are many membraneless organelles. So organelles, by the way, that are not surrounded by a membrane, which can be approximated to liquid droplets. And one clear example is the stress granules that involve the formation of RNA granules, but which also involve proteins in this liquid phase. And so it might have a really physiological relevance. And today we are going to talk about this process, and we have 4 fantastic speakers today. The first will be Nicolas Fawzi, and you can see the title of his talk. And the second is a speaker from Singapore, Jianxing Song. And then we will have Vito Tzurevitz from Case Western Reserve University, and eventually Roberta Cascella from the University of Florence. And so that was my short introduction to the topic. So if you agree, we can start with the first speaker, because I don't want to steal any more time. And so let me just introduce Nicolas Fawzi. Nicolas is Associate Professor of Medical Science at Brown University in Rhode Island, and works in a department of molecular pharmacology, physiology, and biotechnology of the same university. His work focuses on the structure, dynamics, and molecular interactions of large assemblies of intrinsically disordered proteins. A major topic of his work is on LLPS, the liquid-liquid phase separation of RNA-binding proteins, particularly FUS and TDP43, and which are associated with amyotrophic lateral sclerosis, but also frontotemporal lobar degeneration. And his interest is also in protein aggregation associated with Alzheimer's disease. His methods mainly involve NMR spectroscopy and simulation in a combination with biophysical and imaging methods. And so, Nicolas, thank you for joining, and you can start whenever you want.
Thank you so much for the invitation, Fabrizio, and the opportunity to talk today.
Okay.
Let me know if you see my screen okay.
Yes, it is okay.
Great, thank you. Well, like I said, thanks for the opportunity, and I want to thank the Protein Society also. I want to thank President Amy Keating also for, and those who organized the Diversifying Protein Science webinar. I really recommend the talk by Dr. Leroy Jones on interactions with other institutions, so R1 institutions with primarily undergraduate institutions and minority serving institutions. I thought it was a truly excellent seminar. So to talk about liquid phase separation today, as Fabrizio mentioned in the introduction, my interest is looking at intrinsically disordered proteins and NMR spectroscopy. And I want to start off with the most important slide, which is really thanking the people who did the work. And so we had a great group of students and postdocs and some former students, and you'll see work from them. And I'm always looking for postdocs and graduate students. So if you're interested, please definitely contact me if you want to learn NMR spectroscopy and apply it to these problems of protein phase separation. So as Fabrizio mentioned, it's long been known that cells have, membrane-bound organelles, mitochondria, nuclei, but of course, what's become prominent recently is that cells have these intrinsically disordered, these liquid assemblies known as membraneless organelles. And there are many of them, and perhaps the best known is, of course, the nucleolus. But what we're going to talk about today are some proteins that are found in stress granules and other RNA and protein foci in the nucleus. So what do these proteins have in common? Well, unfortunately, many of them are associated with neurodegenerative disease. So RNA-binding proteins with these intrinsically disordered regions that form membraneless assemblies, both inside cells and in vitro. Many of them are associated with protein aggregation diseases that affect neurons. And what do these assemblies look like? Well, neurons are unusual in their need for molecular transport across long spaces. So the length of an axon might be up to 1 meter long, and how do these cells accomplish transport across these things? How do RNAs get trafficked to areas for local translation? Some of them seem to involve these mRNA transport granules where proteins appear to be phase separated with RNA. So the question that we try to look at in the laboratory is, how do these granules assemble? And so there might be a multitude of different types of contacts that form these assembled structures, from RNA interactions to RNA interactions with RNA-binding proteins. But also there's contributions from these structurally disordered proteins interacting with RNA, or perhaps interacting with other proteins or with themselves. But this kind of picture leaves an important question: is where does any of the specificity in these interactions come from? And so one area that we wanted to try to answer this question was in the RNA binding protein known as heterogeneous nuclear ribonucleoprotein A2, or HNRP-A2. So HNRP-A2 is known to have an important role in trafficking RNAs in neurons and oligodendrocytes, where the neuronal or oligodendroglial process is long, and an RNA granule seems to form around HNRP-A2 so that it can traffic specifically bound RNAs. The components of these HNRP-A2 transport granules have been known for many years and include other HNRP proteins, so similar RNA binding proteins, some of which have these disordered domains, others which have RNA binding domains, and some other multi-domain proteins. And together, these are thought to make up the RNA granule. But the question is, many of these studies were done before the realization that these RNA structures, these granules, may in fact be phase separated. So how does liquid-liquid phase separation play a role in these structures? And where does any of this specificity come from in liquid phase separation? So what we wanted to do was ask, how do these different components in HNRP-A2 granules interact? So this work was all led by Veronica Ryan, a talented graduate student who just defended her thesis and started a postdoc at NIH. And so what we're going to look at is, how do these HNRP-A2 proteins interact with themselves or with these components of these HNRP-A2 granules? And then how is that, is there any specificity in these interactions? And we'll compare these interactions with HNRP-A2 to interactions with the FUS protein that's a related RNA binding protein that's not found in these HNRP-A2 granules. So Veronica did a tremendous amount of microscopy experiments in vitro, where she's asking, if we make droplets of this HNRP-A2 protein or its low complexity domain, which we had described in previous publications, we ask, do other components partition into these assemblies? And what you see here are green HNRP-A2 droplets. And if something partitions, something that's we label red, you would see a signal here, and you'd see yellow droplets on this side. And so if we mix in HNRP-A2 labeled in 2 colors, you can see that we can get yellow droplets so that these things mix together. So co-phase separation is indicated by something here in red and an overlay, a merge, showing that we get both components in at the same time. So what we asked was, how do these components of known components of HNRP-A2 granules interact with HNRP-A2? And what we found is that HNRP-A2 can readily phase separate on its own. Neither of these 2 components of the granules phase separate on their own in the similar conditions. But if we mix these proteins, this tumor overexpressed protein, which is a large heat repeat protein, its first domain, which is known, or which was thought to interact tightly with HNRP-A2, we can see that it enters into these droplets formed by HNRP-A2 protein. And we find the same thing for this protein, the prion-like domain of HNRP-F. So both of these things will readily enter into this granule form, or this in vitro droplet formed by HNRP-A2. When we wanted to ask about the specificity, we asked, well, do these same proteins enter into, really any of these in vitro liquid phase separated assemblies? And so what we found is that when we performed a very similar experiment where we mixed the TOG D1 or the HNRP-F prion-like domain into these droplets of FUS low complexity domain, so a similar low complexity domain but with a different sequence, we found that we actually do not see these assemblies, this co-phase separation. So something about the sequence of HNRP-A2's low complexity domain specifies entry of these proteins, while FUS low complexity domain excludes these proteins. So why might that be? Well, we noticed that these low complexity domains, although they are both composed largely of aromatic and polar residues, they actually do have a different sequence composition in terms of their charge residues. And so what we're showing you here is how many of what charge residues are there in these domains. So this 163 residue FUS low complexity domain actually has only 2 charge residues, 2 acidic residues, while HNRP-A2 low complexity domain has arginines in these now famous arginine glycine repeats, as well as some other charge residues. So their charge composition is different. And what we wanted to ask is, is this charge composition important for this specificity of phase separation? So again, we're just showing you here that the F prion-like domain can enter into the A2 low complexity domain, but not into the FUS low complexity domain. You see it's, the partitioning is specific. So what we then tried, well, let's just swap this charge residue composition to make FUS look more like the charge on HNRP-A2 and make HNRP-A2 look more like the charge in FUS. And what we found there is that we were able to swap the charge, the partitioning behavior. And you see that now HNRP-F enters into the FUS that has charge residues and not into the A2 that is depleted in charge residues. And if we do a reciprocal experiment with so we showed by reciprocal experiment that we can specify the partitioning, and we wanted to ask, well, which residues carry the information? And so what we tested was, is it simply the presence of the arginine residues, or is it the net charge in HNRP-A2? And so what we found is that if we altered arginine to glycine, we lose significant amount of the phase separation, but also this co-partitioning. But we can restore the partitioning to HNRP-A2 of the HNRP-F if we simply add the arginine residues back. And so what we find is that arginine seems to be important for specifying this type of interaction between HNRP-A2 and the prion-like domain of another related protein, HNRP-F. So that told us about, one type of interaction that may be happening in the granules, and these are, a simple model of how these might look. But we know that there's other proteins in the HNRP-A2 granules, including this tumor overexpressed gene, TOG, and which had been reported to have tight affinity with HNRP-A2, possibly by binding its low complexity domain. So we wanted to ask, what is the binding actually look like? And so here we used NMR spectroscopy to ask what parts of HNRP-A2 are bound by TOG D1. Where on the TOG D1 surface does HNRP-A2 bind? And we thought that if this interaction was going to be tight, it would be, trivial to look at. But what we actually found is that the interaction was weak, and we had to use a technique called paramagnetic relaxation enhancement NMR to observe our interactions. But what we did find is that the interactions are distributed across this heat repeat surface and involve both the faces of the helices as well as the loops. Importantly, this affinity was significantly lower, 1,000 times lower than a reported affinity in the past. And we think that this might be due to the complications of using phase-separated proteins when measuring affinity and using, for example, surface plasmon resonance. So it's important to consider in your experimental design, how does a multivalent assembly alter what you'd expect to see in terms of your binding affinities. So we find that, in fact, although TOG is able to partition into these droplets formed by HNRP-A2, it doesn't do so because it has a tight affinity, rather it has a relatively weak affinity. So what we're able to see here is that there is some specificity for partitioning, in that these transport granule components that are known to associate with HNRP-A2 in cells, they can co-phase separate with HNRP-A2's low complexity domain in vitro, but not with the low complexity domain of another RNA-binding protein. That seems to depend on, for HNRP-F, the presence of these arginine residues, while for TOG-D1, it seems to interact via the loops and as well as its face. And so, to revise this model of phase separation, or of HNRP-A2 droplets with phase separation in mind, we suggest that these interactions are weak, multivalent, and dynamic, and we wanted to see, how are these droplets assembled? So in addition to assembly, these RNA granules are also disassembled at sites for local translation. And so how does this happen? It seems that these HNRP-A2 complexes with their target RNAs reach a certain site in the end of the oligodendrocyte or neuronal process, and there HNRP-A2 and some other components appear to be phosphorylated by Fin to turn on local translation. So the question is, how does this kinase actually dissociate these granules? So we wanted to see, can we understand a possible mechanism? We had shown previously that HNRP-A2 can undergo phase separation stimulated actually by one of the domains of Fin kinase. So the SH3 domain of Fin kinase is able to actually induce phase separation of HNRP-A2 by a simple addition in a test tube. So although this domain on its own is, well studied for history of protein science, perhaps, we found that by forming weak interactions with HNRP-A2 could actually stimulate the formation of these liquid droplets of A2. And it seems to do that by binding on two surfaces of A2. So we know that Fin kinase has domains that can readily interact with phase-separated forms of HNRP-A2. So we wanted to ask, well, how does this phosphorylation of HNRP-A2 that's been long described actually alter the phase separation as well as the interaction with these other granule components? And so what we found is that if we phosphorylate the low complexity domain of HNRP-A2, we can see that it still forms these droplet assemblies, but TOG-D1 and HNRP-F prion-link domain, they no longer go into these droplets. And so the question is, why does that happen? So it's exciting that the protein is no longer able to phase separate or no longer able to recruit these components, perhaps providing a model for how granules disassemble at sites of local translation. But the question is, well, is it simply an effect of the change in the charge residues that phosphorylation imparts on the protein? And so we tested this by making phosphomimetic forms of HNRPA2, we replaced serine residues with glutamic acid. And what we found was that all of these forms of HNRPA2 could still phase separate or aggregate, but they would all still recruit the F prion-like domain or the TOG-B1. So it seems to be a special feature of tyrosine phosphorylation of HNRPA2 low complexity domain that results in the rejection of these components into these droplet forms. So altogether, what our in vitro results suggest is telling us about how these pairwise interactions between the different granule components may look. And so instead of a tight interaction with HNRPA2 prion-like domain and the TOG protein, we find weak interactions. We also find weak interactions with HNRPF. And we find that the kinase, tyrosine kinase FIN, which is known to be important for initiating local translation, may dissociate the granules by disrupting interactions between the phosphorylated HNRPA2 and the other granule components. So I don't have too much more time, but what we've been trying to do in addition to this work has been trying to see how do these interactions between these low complexity domains actually look like in atomic detail or residue by residue detail. And so what we've done recently is try to look at how do these tyrosine rich regions of the protein FUS interact with the arginine rich regions of that same protein to form these droplet structures. And so a student in the lab, Anastasia Murthy, who recently graduated, has been able to look at, using NMR, how do pieces of different pieces of FUS interact with each other by using differential labeling, isotopic labeling. And so there's many work showing that arginine and tyrosine are particularly important for phase separation of FUS. So we wanted to ask, when we form these droplet structures that contain both arginine containing regions and tyrosine containing regions of FUS, do we see contacts between those regions using nuclear Overhauser effect spectroscopy? And what we find is that we actually do see contacts between these amino acids. We also see many other contacts. So many residues seem to form contacts. We see NOEs not just from one residue type to another, from tyrosine to arginine, but we also see interactions involving glutamine and serine, as well as tyrosine. And so when we look at these contacts using simulations done by our collaborators in Jitane Middle's group, we find that arginine can form interactions with tyrosine, but also many other residue types. And glycine also forms a significant number of contacts. And so our model suggests that many different residue types form contacts and that they can have many different modes of interactions, from cation pi interactions with the arginines and the tyrosines to sp2 interactions involving sp2 hybridized groups, as well as even hydrogen bonding. So we have many different modes of interactions that we think contribute to phase separation of these disordered proteins. And just very briefly, we've been trying to expand this work during this pandemic time to look at how does this nucleocapsid protein of the coronavirus package its genome. And we notice that nucleocapsid protein, of course, it binds the RNA to form the center of the viral capsid. But what we wanted to ask was, how does this packaging actually happen, and might phase separation play a role? And I just wanted to highlight this team of students and former technician in the lab who conducted this work. And what they were able to show is that this protein, the nucleocapsid protein of SARS-CoV-2, readily phase separates, and that a prion-like domain at the N-terminus and joining the two folded RNA binding domains are actually important for robust formation of these phase-separated assemblies. Furthermore, even the formation of phase-separated assemblies by host proteins, human proteins, can be the coronavirus nucleocapsid protein can enter these assemblies. So these disordered proteins are seeming to specify interactions with each other, and what we need to understand is how can we figure out which contacts are important for the structures, which these are ongoing projects in the lab. So I just wanted to thank you again for your attention, and really thank again all the people in the lab who made this work possible. Thank you again.
Okay, thank you. Thank you, Nicolas, for this very inspiring talk. And we have lots of questions already. So if you want to type your question, you can use the question and answer dialogue on your Zoom platform, and you can just type your questions. For any issue related to logistics, then you can use the chat. And we have already 9 questions, 10 now, and let's see what we can do. For example, Einaf Tayeb Flingelman asks, do you see the same behavior in physiological buffer? And if not, how relevant the conditions you are testing here are for LLPS are for the cell physiology and pathology?
So these are certainly done in vitro, and what I didn't have time to talk about is work done by many collaborators looking at these assemblies in cells. And I think it's important to state that these are just models of how these interactions might look. But if you get lots of different of these proteins in vicinity of each other, we think that these are good models for how these questions or how these proteins may interact. And so we try to use physiological buffers and find that this phase separation is robust. It can happen at physiological salt. And interestingly, these proteins, these HNRP proteins, are highly concentrated, among the most concentrated proteins in cells. And so even micromolar quantities are actually found inside cells.
Okay, and we have another question from Anindita Mahapatra. I'm sorry if I don't pronounce your names correctly. How were the charges swapped between the two LC proteins?
So there we just made mutations in the sequence of the proteins and tried to keep the sequence composition and the distribution of the charge residues similar when we did the swapping. So we essentially kept the order and spacing of the charge residues to mimic one protein's charge with the other.
And a question from Ursula Stokai: is Fin involved in the disassembly of protein condensates other than those that contain RNPA2?
You know, that's a good question. I honestly do not know the answer to that. And I think this is things that need to be revisited when we look at what's happening for many of these transport granules. So I know that there's lots of work going on right now looking at many other transport granules in neurons, but also in development. And I think that these questions need to be answered. So I cannot answer the question.
And then a question from Sonia Longhi. First, she says, many thanks, a great talk, Nicolas. Were NMR spectra recorded on samples consisting of both the diluted phase and of the condensed phase? So
Anastasia did collect experiments on the condensed phase as well as a 2-phase sample. So all of the detailed NOE-based measurements that can let us see interactions were done on samples that have just the condensed phase. So they're made from a really large sample that we only take the 15 milliliters or more and only look at the condensed phase that we can look at. But in work that she published last year, she showed that we can see the same fingerprint in a two-phase system where we have droplets that are formed spontaneously, and we see the same spectral signature when we have two phases present. So we can see the condensed phase that's formed spontaneously.
Okay, and maybe we should stop here. There are other 15 questions, maybe more. So if Nicolas, you want to answer to these questions, you can just, we go on now, and you can just type your answers on the questions and answers box, okay? And so we can then go ahead and let me introduce Jianxing Song, who is an associate professor at the National University of Singapore. He works in the Department of Biological Sciences of the same university. His main research interests are on protein biophysics approached with both experimental and computational methods, molecular mechanisms of protein aggregation and its associated deleterious effects in both disease and aging. His favorite proteins are intrinsically disordered proteins as well as membrane interacting proteins, and is also interested in drug discovery. So I just, yeah, go ahead.
Okay, thank you very much for the invitation and also introduction. Can you see?
Yes, we can.
Okay, so for my talk, I'm going to talk about something like a new function of the ATP. So basically, here is a list of the section I'm going to present quickly. But I think thanks to the great talk by Nicolas, I can actually, with his talk, and then I can jump over some slides. So basically, at the very beginning, people think all the protein going to fold into the well-defined structure. But recently, actually, we just started to realize, actually, for protein sequence, you can roughly divide it into two categories. One is high complexity one, another one is low complexity one. In fact, only a small portion of the high complexity sequence eventually will fall into well-defined structure. For the low complexity, which a portion will become like a so-called intrinsic disordered protein. Actually, from both like, categories, a large portion of a protein will become aggregated, and then we'll see aggregated protein is really a big problem for diseases. So for the well-structured protein, actually just a couple, I think 1 week ago, actually, Google just announced they have the program which can predict the structure with very high accuracy. So now I think there's another category of proteins, so-called intrinsic disordered protein. So here is the classical function for the intrinsic disordered protein. But recently, people found out the intrinsic disordered protein has another very, very important function. It's called like phase separation. And then actually, Nicolas already gives a very nice introduction. So actually, many years ago, more than half a century ago, people found out the folded protein also can phase separate. But for folded protein, phase separate at very high concentration, like 100 to 400 milligram per mole. But for the intrinsic distorted protein, actually, the concentration needed for phase separation quite low. For example, the one protein actually fast we are working on, the full length, you just need 1 micromolar concentration and then can phase separate nicely. And then the reason why the intrinsic disordered domain can phase separate so effectively is because people propose this due to the dynamic and multivalent interaction. And then, of course, the big reason why liquid phase separation becomes so important, actually, Nicolas also introduced, is actually responsible for the formation of large like the list of so-called membranous organelles. So And then there's another issue, as I mentioned, actually, for both folded protein and the intrinsic disordered protein, a large list of them eventually will become aggregated. This is a pioneer work by Professor Chiti and also Dobson. Actually, turned out, okay, most people think, well, if protein aggregated, and then we are the need to the loss of a function, that's all. But it turned out actually the extensive studies suggest actually protein aggregation causing need to the diseases, not majorly due to the loss of function. Actually, instead is gain of the new toxicity. So this is actually most well-known diseases, is all neurodegenerative diseases related to protein aggregation. Also, people recently found out that aging, probably aggregation is also the core reason for the aging. Actually, it turned out before people think like the E. coli, single cell organism, no aging. But just I think around 10 years ago, the scientists found out even for E. coli cell, there's aging. The only sign, biomarker for aging of the E. coli is just the protein aggregation. And then why the liquid phase separation actually connected to diseases actually Professor Chiti is already introduced, because in a droplet, actually the local concentration in the droplet is around 50 to 300 times higher than the environment. So for protein, like we are working on, and Nicolas also working on FAST and the TDP43, such protein already intrinsically prone to aggregation. And then once concentration enriched in the droplet, and then definitely it's so easy to transit into the aggregation. So this is why the people believe also the liquid phase separation is a bridge to the diseases. And then, no, let's look at another issue is whatever the protein or even all the life form on Earth is linked to water. But water is a very mysterious molecule. Actually, people think even though we are still not really understanding what is water. One example is our discovery many years back in 2005. We found that actually all insoluble protein we tested so far more than 60, actually even included the most hydrophobic transmembrane fragment in nature without lipid can be dissolved in pure unsalted water. And then we can look by CD and so, but once you add the salt precipitated red or white. So this is actually a lot of people and scientists still not believing unless they tested it. And then also we collaborate with some like hardcore simulation people, even use the quantum derived like the parameter to do the simulation, still cannot really simulate this phenomenon, because if you need to use the computation to understand the water, you need to consider so-called polarization effect. This needs the quantum-based parameter. So far, it's still very challenging. Probably in the future, if there's a quantum computer available, possible. But anyway, another the issue proteins going to encounter in cell is actually cell is extremely crowded. So usually it's 200 to 400 milligram per mole in cell. And then if you talk about this, actually people concluded in such a crowded environment, to keep the protein soluble, one very important property is electrostatic interaction, because this is the long-range interaction, where the hydrophobic interaction is the short range. So now I think this go to our the topic today. So based on our study, and then we think probably ATP has integrated the property of the like something like the pure water. On the other hand, also like the salt, because on the one hand, the salt can cause the aggregation of insoluble protein, but it's required for function and for some folding of the protein. But the ATP seems to integrate the property of the pure water and the salt. Okay, so of course, ATP is such a common molecule, everybody studied that. And then before, the so-called classical function is all about the chemical reaction related to so-called high energy bonds. And then they're responsible a huge variety of the function. But usually people found out for such category of function, the macromolar concentration of ATP is sufficient. But so for all the living cell, ATP concentration actually pretty very high, from 2 to 12 millimolar. So a fundamental question is why? Because you know ATP, unlike the sodium chloride, is just the diffusion and then environment very concentrated by diffusion, and then cell can have the very high concentration. But for ATP, you need a very complex machinery to generate. And then fundamental question is why cell invest so much energy to maintain such a high concentration? Currently in the textbook, they propose, because this is, you need the free energy gradient between the ATP and ADP and AMP. But anyway, that's a very important question to ask is, at the millimolar of ATP, well, whether there's some novel function. So for this question, actually, the very important paper is from Hammond's group. So based on the structure, chemical structure of ATP, you can see this is a hydrophobic, this is a hydrophilic. So this kind of a molecule can be called a hydrotropic molecule. So in this paper, Hammond's paper, and then they proposed actually with concentration larger than 5 millimolar. Actually, ATP, the hydrotropic, the purine ring can cluster over the hydrophobic surface and then eventually inhibit or even dissolve the amyloid fiber or aggregation. And then our further study suggests that probably ATP's function, novel function, even go beyond the hydrotropic molecule. So we found out here is what I proposed. Actually, ATP also can be considered as a bivalent binder. Why? Because here the purine ring actually is aromatic. Actually, Nicolas already talked. Once it's aromatic and then can form the peptide interaction or peptide-ion interaction with amino acid like arginine or lysine. And then on the other hand, the triphosphate group is highly negatively charged and then can establish the electrostatic interaction with also arginine and lysine. This is still understandable. But there's another the property I just realized could be important is not really appreciated before. So according to a paper published from a Japanese scientist here, so he found out actually, okay, whatever in solution, there's a box solvent, and then the supposed water in the box solvent can move freely, and then around some surface like the protein, amino acid, even the ATP, there's some constant water, usually called hydration share. So for such kind of water, the motion compared with bulk solvent, bulk water should be slower. But ATP very unique. So by the so-called like dielectric wave spectroscopy, he found out over the triphosphate the chain, there's another share of the water. It's called hypermobile water. Around this region, actually water moving even faster than the bulk water. So this is so unusual. This is actually, we recently found something very unusual. We believe it's due to the presence, so-called the hypermobile water. We are seeing quickly. So, okay, first, let's talking about, look at the so-called the bivalent band. And then this way, I'm going to illustrate this with FAST. FAST, very big one, but here, because of time limitation, I just focused on the C-terminal, so-called the RGG rich domain. So RGG rich domain, very short one, 156 or something, and then contain 25 like arginine. So for this one, if just the RGG domain, fast RGG domain cannot undergo fast separation, even with protein concentration reach 1 millimolar with salt to 500 millimolar. But once you add the ATP, you see, we are induce the fast separation even at 0.2 millimolar ATP, and then at around 1 millimolar ATP, and then fast separation reach the highest. And then once you further add the ATP, and then droplet started to be dissolved. And then we also use the same method as Nicolas introduced, use AMR. And then we make assignment, and then we found the shift mostly centralized around the arginine as a nation. But unfortunately, because the fast contain, RGG domain contains so many arginine, 25, we cannot really, the completely for sure to say ATP band which residues. But anyway, we found that actually ATP and then oligonucleotide acid actually behave the same, but the oligonucleotide acid, the affinity much higher, both for induction and dissolution. ATP, the capacity lower. So we propose actually both like nucleic acid and ATP target arginine and lectin. But as I told you, because the FAST is so contains so many arginine, cannot really tell. Now we just recently moved to the prion domain of the TDP43, because for this one around 150 amino acid out of this region, the 150 amino acid, only 5 arginine there. So this way, we can use both AMR and also mutagenesis really to map out the ATP is going to bind which residues. And then we found out this one, we adjust the concentration and then make it without ATP, no fast separation. And then we add ATP and then fast separate at both pH 5 and also the pH 7. And but again, if you further increase the concentration of ATP and then started to dissolve, so the same by like phasic modulation. And then we use AMR, we can immediately to find out ATP is going to interact with which residues. And then once we identified such residues, we can mutate it. It turned out we found out ATP only bind to the arginine and the lysine over the intrinsic disordered protein. No other residues significantly affected by ATP. But it turned out actually, we found out actually even arginine and lysine, but arginine interaction between ATP and arginine much stronger than the ATP with lysine. The reason is quite simple, because chemically, you see, because arginine have the unique denucleonize the double bond, and then can have the both like peptide interaction, the cation ion interaction with the pure run of ATP. But for niacin can only have the cation ion interaction. This is why arginine ATP interaction is so strong. So turned out we found out actually we therefore propose the mechanism. So why like in a normal cell, even neuron with ATP concentration at only 3 millimolar, under the physiological condition, 3 millimolar sufficient to dissolve the fast separation of the TDP-43. Because recently, Cleveland's group in UCSD demonstrated fast separation of the TDP-43 alone without involving the form of the stress granule is sufficient to trigger the toxicity to cause ALS. So, but why under the normal condition, no fast separation? We believe at least one very important molecule to keep the fast separation inhibited in the normal neuron is because of the presence of ATP. But once become aged or TDP-43 concentration overexpressed, and then the ratio between ATP and the TDP-43 become reduced, and then there will be fast separation and further aggregation of amyloid fibrillation, and then eventually lead to ALS. Okay, so now I think from our study on FAST and the TDP-43, you found out the ATP can trigger the biphasic modulation by two mechanisms. One is called the direct mechanism. Another one is indirect mechanism. So for FAST one is called the direct mechanism, because the FAST contained 150 residues about the same, contain 25, and then just the simple ATP bivalent binding is sufficient to make a big like the dynamic multivalent like cross-linked like the complex, which behavior manifests as a droplet. But for TDP-43, only 5 residue arginine within 150 amino acid intrinsic disorder prion domain. Only ATP cross linkage not sufficient, therefore need the other driving force. One is very important, one is what actually Nicolas group demonstrated, identified before, is the demyelination of the helical, like hydrophobic helical. So ATP actually behavior as an initiator to trigger such interaction or enhance, and then to trigger the fast separation. So turned out our results suggest actually ATP the most only bind to the arginine. Therefore, ATP so looks like ATP is in addition to the covalent arginine methylation, ATP is probably a dynamic mediator for the arginine content in intrinsic disordered like regions. Okay, now let's move to another function of ATP. We found out that ATP can inhibit amyloid fiber formation of the RNA binding motif. So this one actually we also look at the FAST, because in addition to the intrinsic disorder, the FAST contain one well-folded domain called the RNA recognition motif. And then we found that the ATP can bind to the pocket actually, which is used for binding to the nucleic acid. And then even for FAST binding without affect stability, but sufficient to kinetically inhibit the amyloid formation. And then moreover, we found that ATP can also bind to the TDP-43 RNA domain. And then for bind to the TDP-43 first RNA binding motif, and then can even enhance the stability, but anyway, can also inhibit the amyloid formation. Recently, we also found that another the RNA binding motif, which is totally different from the RNA binding recognition motif, all helical, also can bind the RNA. But you see such domains, their binding affinity pretty low. It's around several millimolar for dissociation constant, very different from the kinase. Usually the ATP's binding affinity to kinase for KD is around like at least macromolar, some even for rich nanomolar. So if you look at the interaction critical for such kind of a complex, weak complex, you see the pi-cation ion interaction or pi-pi interaction between the purine ring of ATP as well as with aromatic amino acid in protein. Also this is a pi-pi, this pi-cation ion, and also there's some electrostatic interaction as well as a hydrogen bond. So now I'm going to briefly show you something we still cannot completely understand the mechanism, because you know for the air length, it's probably air length is cell is the most crowded like cells. Because in length, actually, the like crystalline concentration can reach 400 milligram per mole for human. In fish, can reach 1,000 milligram per mole. But unbelievably, even at such a high concentration, at least also there's no turnover or replacement for such kind of a crystalline. So that means from the birth of the human being to the death, there's no replacement, no turnover. And then for all the life being, so crystalline need to be soluble. If anything like aggregation occur, and then we need the eye diseases, one is called cataract. And then also people found out for alpha S crystalline, even at 400 milligram per mole, it's still monomer. Although there's a lot of biophysics study shows actually the monomeric interaction is attractive, but still remains a monomer. Another very, very mysterious issue is the islands, there's no, almost people think there's no metabolic activity. There's no mitochondria, but still the ATP concentration very, very high. Is 3 to 7 millimolar, even higher than neuron. So why the islands keep such a high concentration? Even though people still not knowing clearly where such ATP from. There's a different speculation, but anyway, here we care is any effect of ATP for the gamma S crystalline. And then we first express the protein, we use AMR to detect at the very low concentration, like just 100 macromolar. And then another one is 100 millimolar. This one is 100 macromolar. This is, I think, it's around something like the 10 millimolar. And then
1 minute, 1 minute.
Okay, and then we found out actually no binding. But once you look at the concentration dependence, you found out ATP can antagonize the concentration dependent destabilization. So therefore, based on this, we propose ATP actually is going to interact with the hydration share of the protein to make it more stable. No, this is quickly, just one or two slides. The pandemic, actually, the Nicolas already showed you there's a phase separation. And then what we found is ATP also modulate the phase separation between the N protein and the RNA and the interaction. Also, we found out actually ATP is going to bind to the RNA binding domain of the N protein. And then we propose probably this SARS coronavirus 2 is very smart. They use the ATP in the host cell to promote their life cycle. And then this is just a quick summary. So since ATP at the minimal concentration, that's a novel category of the function. So basically, the major function for ATP is to control the protein homeostasis. But other function also possible, like the modulator probably SARS infection. And then also there's one phenomenon is all the neurodegenerative diseases, actually the risk increased with aging. And then corresponding to the observation, ATP concentration reduced with aging. And then this is the student and the postdoc who did the research. Finally, thank you very much for your attention. Yeah, okay, thank you.
Okay, thank you, Professor
Thank
Song.
you. Yeah.
Time for a couple of questions. Ellen Jaff asks, how does the interaction of magnesium with ATP impact your point?
Sorry?
How does the interaction of magnesium ions with ATP impact your point?
Oh yeah, actually, because actually we always put the mechanism chloride because it's going to form like the chemical bond between the second and the third like phosphate to stabilize the high energy bond. So this is what we did because our reaction related to no chemical reaction. So this is actually Hammond also at the very beginning that put the mechanism chloride to stabilize the equimolar.
And another question from Eric Martin, perhaps I missed this, but is there a reason to believe that it is the aromatic component of ATP that interacts with the positive arginine-lysine groups?
Oh yeah, indeed. Because it's a bivalent interaction. So aromatic ring, purine ring is extremely important. It's responsible to establish the interaction with arginine side chain by pi-cation interaction. On the other hand, triphosphate chain also interact with another arginine side chain. So this is called bivalent binding.
Okay, so I'm sorry, but for the sake of time, we have to go ahead with the schedule, and there are many more questions for Professor Song. So if you can stay and type your answers to all your questions, that would be appreciated. Let me.
Okay, thank you. Thank you.
And let me go ahead by introducing Professor Witold Tzurevitz, who is a Robert Bennett Professor of Neurological Research at Case Western Reserve University in Cleveland, Ohio. He works in the Department of Physiology and Biophysics of the same institute. His research has for a long time focused on molecular basis of prion diseases and other neurodegenerative disorders associated with protein misfolding. Recently, he became interested in LLPS of proteins involved in neurodegeneration, such as tau and TDP43, publishing works that have been very inspiring, similarly to other speakers today. So thank you, Vito, to join us, and you can start whenever you want.
Okay. Let me share my screen. Let me activate the pointer. Okay. So first of all, I would like to thank Fabrizio for organizing this meeting, and of course Protein Society, and in particular LACA, for putting everything together. It's a great meeting. I greatly enjoyed the presentations. What I want to talk about is it's about the link between phase separation and aggregation of proteins, neurodegenerative diseases. And as you know, as has been already alluded to in previous talks, I mean, phase liquid droplets play major physiological role in great many biological processes, and that has been addressed in the previous talk. But you know, what was kind of, I think, exciting development in the field was the finding that many proteins involved in neurodegenerative diseases that aggregate and forming those plaques, which are toxic and so on, that they have either they are intrinsically disordered or have low complexity domains, which are known to drive phase separation. So kind of this suggested that there might be a link between those two processes, phase separation and formation of those aggregates and eventually amyloid plaques, which and those plaques, of course, you know, this is kind of by far not complete list of proteins involved in different neurodegenerative diseases that have been shown to undergo liquid-liquid phase separation. But really the mechanistic link between phase separation and aggregation or formation of amyloids is really very, very poorly understood. So here I want to focus on this problem, just giving you an example with studies of two proteins. One is tau, and if time permits, I'll also talk a little bit about TDP-43, but in a little bit different context. So few quick words about tau. Tau is a microtubule associated protein that kind of exists usually in normal cell. There is a balance between tau form associated with microtubules and the free tau, but this equilibrium is shifted strongly towards the bound form. But when something goes wrong, what we see is that large number of tau molecules become dissociated from microtubules. This is typically induced by phosphorylation of certain residues, and the free protein starts to aggregate, forming those kind of different types of filaments, which really are amyloid fibrils, but they come in different morphologies, and they form eventually those what is called neurofibrillary tangles. And those tangles have been observed in great many of diseases, ranging from Alzheimer's to Pick's disease to frontotemporal dementia with Parkinsonism 17. So I want to talk a little bit about this link between phase separation of tau and aggregation. Now, very, very quickly about the structure of tau. It has a domain structure, but there is a so-called repeat region of for repeat units, which are 31, 32 residue long, and though this repeat region is known to drive the aggregation fibrillation process. There is a proline-rich region, and there is kind of an N-terminal insertion. What is really important in the context of what I'm going to talk about it is that due to alternative splicing of mapped genes that encodes the protein, there are 6 different isoforms of tau that differ with respect to the number of those repeats domains, and also with regard to number of those N-terminal inserts. So this will be important. Tau has been already a few years ago, that was reported by several groups that it can undergo under different conditions in the test tube phase separation. Also, there are reports that it can undergo phase separation in different type of cells. It can form kind of complex conservates in the with RNA or with other polyanionic polymers such as heparin. This has been shown by Hans group. Then it has been shown that also K18, that this repeat region fragment can also undergo phase separation, but only under kind of the conditions of pretty high, very high protein concentrations. And what was unusual is that it requires very, very long time to form those droplets, which is highly unusual. Kind of, and then 2 years ago, Susan Wegman has shown that phosphorylated tau in the presence of crowding agents such as Ficol or polyethylene glycol can also form those nice liquid droplets. But what Susan Wegman reported, that this is specific to phosphorylated protein in her hands, and non-phosphorylated protein had no ability to phase separate. But when we kind of started working with it, what we found that really in the presence of crowding agents such as polyethylene glycol or dextran, ficor, one doesn't need to have phosphorylated protein. And this is mostly the work of Solomia Boyko, what I'm going to talk about. She's a graduate student in the lab. But so what we found that it undergoes easily very robust phase separation in the presence of crowding agents. And what was important, that this was kind of promoted by low salt concentrations. When we kind of increased salt concentrations, we required more and more polyethylene glycol. But this could be observed even at physiological tau concentrations, even at 2 micromolar concentrations. So kind of, this indicated to us clearly that what is really driving the process are electrostatic interaction, because this is extremely strong dependence to salt. And of course, we could kind of, support this conclusion by using hexandiol, which is an agent known to disrupt phase separations that occurs, is driven by interactions other than electrostatics or charge-charge interactions. And in this case, hexandiol wouldn't really be able to do anything to phase separation. So kind of that, prompted us to look a little bit more in detail into how is it happening. And when looking at the plot of number charges per residue, which is here, kind of as a function of residue, one can notice right away that there is a kind of polarized distribution of charges. N-terminal part of the molecule is highly negatively charged, and the middle part and C-terminal is just rich in basic residues. So then we kind of try to play with this polarity to see whether this plays any role in this. And to cut the long story short, one can really monitor phase separation by simple turbidimetric assay, but of course one needs also to confirm it by just the optical microscopy, looking, verifying that the droplets are present indeed. But when we started playing with and making different constructs, which we kind of gradually deleted certain regions of the protein. For example, when we delete this N-terminal negatively charged domain, we completely lost the ability for the protein to undergo phase separation. So kind of from all those experiments, what we kind of led us to the model to the conclusion that probably the phase separation is largely driven by attractive electrostatic interactions between this negatively charged N-terminal part of the protein and positively charged middle and C-terminal part of the protein. But to just prove it even more conclusively, we just separated those two parts of the proteins. Of course, none of those parts was able to undergo phase separation on its own, but when we mix them together, we could see a beautiful phase separation. So this led us to this conclusion that our model, that's all driven by attractive electrostatic intermolecular interactions between negatively charged N-terminal part and positively charged middle and C-terminal regions. But if this is the case, we ask the question, which is an important question in the context of tauopathies, which are kind of diseases collectively known as those diseases associated with aggregation of tau, are collectively known as tauopathies, and there are many of them, as Alzheimer's disease, Pick's disease, and so on and so on. Then kind of if our model is correct, then those mutations really shouldn't affect that much the ability of the protein to undergo phase separation. Many of, and we focus on 3 of them, but many of them, this is more comprehensive list, don't change the charge. And even if some of them change the charge, it's only one residue. And it seems to be the ability of the protein to undergo phase separation seems to be a global property of the entire molecule. So, and especially we're kind of prompted by some suggestions that actually those very same mutations strongly promote phase separation. It has been suggested in a number of papers. But when we did the experiments, what we found, okay, so how we do the experiments? The easiest way to look at the propensity of a given protein under given experimental condition to undergo phase separation is to measure so-called saturation concentration. So below saturation concentration, protein will exist only in the bulk phase. There will be no phase separation. And then at saturation concentration, proteins will start phase separating, and the higher the concentration, of course, the more protein will be separated. But within the droplet, the concentration of the protein which remain constant. And you can measure it by different methods, including turbidimetric assays. And what we found, consistent with our model and prediction, that none of those mutations had any effect on the ability of the protein to undergo phase separation, at least under those conditions, which is in the presence of 10% PEG. So because they had identical saturation concentration. So, but the question was, maybe it's only happening under those particular conditions with this concentration of PEG and this salt concentration. So we kind of looked at it, across different type of experimental conditions, constructing so-called phase diagrams, protein concentration versus salt concentration of protein concentration versus PEG concentration. Those green boxes here there is phase separation, gray boxes, no phase separation. You can see mutations have absolutely no effect on the propensity of the protein to undergo phase separation, which is kind of consistent with our electrostatic interactions model. But then we ask the questions, and mutations, of course, play a major role in the disease. One thing I should say here, that in order to form amyloid fibrils with heparin, we require anionic cofactors, such as heparin or nucleic acids. And then, so we are kind of asked the question, how really mutations, they might not affect the propensity of the protein to undergo phase separation, but how would they affect the aging of droplets, their maturation into something which is more solid-like? In the absence of heparin, we didn't see any maturation. It was, there were dynamic droplets over many hours. But in the presence of heparin, this aggregation-inducing agents of tau, what we found that for some mutations, actually, there was, especially delta K280, there was extremely rapid maturation of those droplets. And you can see also here, they were losing kind of liquid character, and they will become aggregates. Those experiments were done here. What we are showing here are so-called FRAP traces. We simply bleach certain region of the droplet with laser, and then we look at how fast is the recovery of the fluorescent signal. Of course, the more dynamic the droplet, the faster would be the recovery. So what we found here was that the material properties of those droplets in the presence of heparin are strongly modulated by those pathogenic mutations, with some of those mutations lead to very rapid liquid-like to solid-like transition. So next we ask the question, does it really affect a real aggregation, formation of stable aggregates? Those of you who are not in the amyloid field, so kind of typical aggregate fibrillation reactions, there is always a long lag phase, which corresponds to the formation of the nucleus, and then there is elongation phase. And we can kind of monitor it by simple fluorescence-based assay using a fluorescence dithioflavin T. And when we kind of look at this, what we found that for both for the wild type protein and all those pathogenic mutants we looked at, there was a very, very dramatic increase in the rate of fibrillations under conditions of phase separation as compared to the conditions where we had no phase separation. And this effect, for example, was especially dramatic for delta K280 mutants when in the presence of phase separation, there was essentially completely lost lag phase, whereas under no phase separation conditions, the lag phase definitely would be longer than 40 hours because we couldn't detect it over any aggregation over this time. And we confirmed that those are indeed amyloid fibrils by just verifying it by atomic force microscopy. Those fibrils, those are different magnifications. As you can see, those fibrils are really clumped. What makes sense because everything is happening within the droplets. Under the conditions of those experiments, the concentration of protein outside droplets was minimal. In very, very, very low. So what we concluded from it was, of course, LLPS strongly accelerate fibrillation of tau. This was our number one conclusion. And our number two conclusion was that obviously different types of interactions might be driving LLPS of tau and fibrillation within droplets, because LLPS, as I said, is driven by those attractive electrostatic interactions. But fibrillation, what we know from many, many studies under non-phase separation conditions, that all the action is within this repeat domain, where kind of certain segments, so-called PHFs fragments, those 5 residue segments, play an especially important role. And when we remove those segments, actually protein loses its ability to undergo phase separation. So it looks like those two things, phase separation and aggregation, there's no correlation between a defect of mutation on those two different phenomena. But then we ask a question, are there any unique features of protein fibrillations under the condition of liquid-liquid phase separation? So, when we do the experiments in the absence of phase separation, of course, the reaction kinetics would be strongly concentration dependent, protein concentration dependent. It's a higher order process. So of course, the more protein we add, which is kind of here, it's indicated by the intensity of the color, the reaction will become faster and faster and faster. But what is a unique feature of phase separation, that when we keep increasing the concentration of the protein, everything is dictated by saturation concentration. So what will happen above saturation concentration, no matter how much protein we'll keep adding, the concentration within droplets is not going to change. The only thing that will change is that the number of droplets, of the volume of the protein that forms droplets, would become larger. And this, so our prediction is, well, that if we look at the fibrillation rates within droplets, it wouldn't really matter what is our concentration of the protein. So it would be fundamentally different ballgame that in the absence of phase separation. So then we kind of ask another question, would there be really any kind of important consequences of this phenomenon? So here, to explore it a little bit in greater detail, we kind of turned to, we again took advantage of the fact that what is really important, crucial for aggregation, is the presence of these PHF, 5 residue PHF segments in the repeat domain. And when we remove those segments, the protein completely loses the ability to undergo phase separation. And of course, 2N4R is tau, full-length tau isoform, which we have those 2N terminal repeats and 4 repeats, 4 pseudo repeats in this important region. So it's the longest form of the protein, which is highly aggregation prone. So we ask the question, and what is important, that removal of this delta PHF region does not affect in any way propensity of the protein to undergo phase separation. Because it's the saturation concentration is unchanged. So we ask the question, what will happen if we mix those two proteins together? Which is, because one would expect here, the concentration within droplets of those two proteins, they are totally miscible in droplets. So the concentration of those two total concentrations always be constant. So then when we keep adding higher and higher proportion of this non-aggregating protein, essentially we should see the acceleration of the reaction, because we will dilute the aggregating protein within droplets. And that's exactly what we could see across all those, wild type protein and the mutant proteins. So, this is, those numbers corresponds to the ratio of wild type protein or the mutant to this delta PHF form. When we started at the delta PH form, the reaction becomes slower and slower, simply because we are diluting within droplets the aggregation from isoform of the protein. So first, here it's a kind of a little bit of an intellectual exercise showing something like this can happen, but delta PHF, of course, is not a physiological form of tau. But what is important here in physiological context, that actually it might be of profound consequences, because we will very often will have a mixture of those two different tau isoforms with different numbers of those repeat pseudo-repeat units. So this would be 2n, 4R and 3R. 3R aggregates much more slowly. It's much less aggregation prone. So then when we mix them together, we would predict that the slowly aggregating form should again dilute the concentration within droplets of fast aggregation form, and that we should see slowing down of the reaction as the proportion of this 3R form is increased. And that's exactly what we are seeing. So why is it important? Because the ratio of those two tau isoforms varies across different phenotypes of tauopathies. For example, in Alzheimer's disease, it's roughly equal, but in peak disease is considered to be roughly 3R tauopathy, progressive supranuclear palsy, or other diseases are 4R tauopathy. And we have, of course, familiar forms of the disease which were related to the mutations in the proteins. And those mutations are known to affect alternative splicing. So depending on the mutation, we can have a different ratio of 3R to 4R isoforms. So this seems to be something of importance. And I think if I have so kind of, we ask the question, what factors really control the aggregation, the rate of fibrillation under phase separation conditions? Of course, there will be very high protein concentration within droplets. It is well known. Actually, in the case of tau, our estimation is something what I cannot believe, that it might be as high as around to 1 millimolar. So this would, of course, have a strong effect promoting phase separation. But one shouldn't forget, which is very often totally ignored, that the environment and within droplets is extremely, of course, the dilution effect, we have different isoform, could kind of decrease the rate of fibrillation. But another factor is viscosity, which is extremely high within droplets. We try to estimate how viscosity would affect, just under non-phase separation conditions, how viscosity would affect the rate of fibrillation. But here you can see even at 10–20% of glycerol, which we added here, so those are different concentration of glycerol, which increases the viscosity. There was, very, very rapid decrease in the rate of fibrillation. We couldn't really detect anything, the concentration of the glycerol above 30%. But this is only, glycerol. Keep in mind that the viscosity within droplets, the viscosity even of 100% glycerol is roughly 1.4 pascal second. And within droplets, typical viscosity would be of the order 10 to 50 pascal second. So order of magnitude higher than in 100% glycerol. So this would be a powerful factor that will actually try to prevent fibrillation. So we'll have those kind of, the struggle between those two factors. So it's not necessarily that fibrillation will always result in massive increase in fibrillation rate. Of course, and of course we'll have this dilution effect, which is specific to situations when we have different isoforms of the protein, but the similar phenomenon potentially could be observed in the presence of other proteins. And of course, another big question would be, another factors could be conformational change in the environment of liquid droplets. We know very little about it, how the conformation changes, and it could be that the change that potentially either promotes fibrillation, or it could be the one that inhibits it. So this is something what still needs to be studied. Fabrizio, how much time do I have? Or I
was about to interrupt you. It's you have 1 minute left.
Okay, so I wanted to talk about something else which is related to Dr. Song's talk about kind of how to modulate phase separation in the biphasic way with small molecules, but at very, very kind of not with ATP, but with molecules kind of designed for this purpose that act at a very low concentration, micromolar concentrations. And we came across about around such molecules But I won't be able to talk about it today, but we just published a paper a month ago in Nature Communications. So if anyone is interested, I will refer you there. And thank you so much for listening.
Okay, thank you, Vito, very much for your very interesting talk. And as ever, we have lots of questions here. Boris Zaslavsky asks, what molecular weight of PEG did you use in experiments with tau?
Here it was 10 kilodalton. But we can see those phenomena, you know, with 3 kilodalton, we try to do, but whatever I've shown here, it was 10 kD.
And Eitan Lerner says, thanks, very interesting. In promoting tau LLPS, did you use macromolecular crowders which induce intermolecular interactions? So this was not the question, this is the point. And the question is, did you try low molecular weight crowders such as osmolytes like TMAO? Mm-hmm. If yes, did these induce stabilization, intermolecular interactions between the negatively
this is something what we are thinking about, but we haven't done an experiment so far with those kind of so-called chemical chaperones. No.
Okay. And Ursula Stokai, she says, does the presence of microtubules alter the saturation concentration for tau LLPS? How do the self-resistance compare to LLPS of tau in cells? Okay,
so we haven't done much of experiments in the cells ourselves. There are few reports. There's one Nature's, I believe, neuroscience paper when people were doing it in cells in the context of tau interactions with microtubules. I cannot answer this question because I don't know whether this would affect and to what extent and how it would affect the saturation concentration. I don't know.
Okay, thank you, Witold, very much. If you can also stay and answer to the various attendees' questions, that would be appreciated by them.
Sure.
We have to go ahead, and let me just introduce the last speaker of the session today, Roberta Cascella, who is Assistant Professor of Biochemistry and works in the Department of Experimental and Clinical Biomedical Sciences at the University of Florence in Italy. Her research activity focuses on the mechanism of toxicity of protein assemblies, ranging from protein oligomers to fibrils and involving mainly proteins associated with neurodegeneration, including the amyloid beta peptide, alpha-synuclein, TDP-43. And the title of her talk will be the involvement of stress granules in TDP-43 cytosolic inclusion formation. So go ahead, Roberta, whenever you want, you can start.
Thank you, Fabrizio, and good afternoon to everybody. It's a pleasure for me to be here and to have the opportunity to show my research. Can you see my presentation?
Yes, we can.
Okay, so today I will talk about involvement of stress granules in TDP-43 cytosolic inclusion formation. In 2006, a famous paper published in Science from the group of Virginia Lee demonstrated the involvement of RNA-binding protein in amyotrophic lateral sclerosis and frontotemporal lobar degeneration. They found that intracellular inclusions in the central nervous system of ALS and FTLD patients are formed by the TDP-43 protein. This pathological TDP-43 containing these inclusions was found to be hyperphosphorylated, ubiquitinated, and cleaved at the C-terminal domain. TDP-43 is a multifunctional nuclear protein originally described as a transcription factor, but later found to be also involved in other cellular processes. It is predominantly a nuclear protein, but also shuttles between the nucleus and the cytoplasm, where it is involved in stress granule formation, translation, and other processes. The pathogenic mechanism of TDP43 neurodegenerations seems to start from the cytoplasmic accumulation of the protein in pre-inclusions and inclusions that are hyperphosphorylated, ubiquitinated, and cleaved at the C-terminal domain, together with the loss of the protein from the nucleus. So, in the last year, numerous researchers have tried to understand whether the TDP43 pathology is caused by the nuclear depletion with the loss of protein physiological function, or by the presence of TDP43 aggregates in the cytoplasm with cytotoxic effect. To this aim, we have recently quantified the relative roles of loss of function and gain of function mechanisms and their contributions to the cytotoxicity of TDP43. In this study, we used murine motor neuron-like cells, which are a widely used cell model for ALS pathology. And we transfected the cells with different quantities of plasmid overexpressing human full-length TDP43. And we assessed the presence of the protein by using confocal macroscopy. As we can observe, transfection with moderate amount of plasmid caused a significant increase in TDP43 nuclear levels, without any significant modification of the cytoplasmic TDP43 levels. With a higher amount of plasmid, we observed the progressive accumulation of the protein into the cytoplasm, together with a loss of the protein from the nucleus. In particular, transfection with 10 micrograms of plasmid showed a redistribution of the protein from the nucleus to the cytoplasm, simulating the pathological condition. With larger amount of plasmid, we observed a progressive decrease of total TDP43 levels, probably due to an autoregulation process or by the clearance pathways activated by the cells. We then assessed the mitochondrial functionality of the cells by using the MTT reduction test, which is a widely used indicator of cell viability. We can observe that the overexpressed protein caused a significant reduction of cell viability, the degree of which increased with the quantities of plasmid. Similar results were obtained from the analysis of the caspase-3 activation. But the toxicity that we observed can result from both the nuclear depletion or from the cytoplasmic accumulation of the protein, or from both mechanisms. So, to dissect the two contributions of loss of function and gain of function mechanisms, we used an approach to analyze separately the loss by silencing the expression of endogenous TDP43 with small interference RNA, and the GOF by inserting TDP43 aggregates in the cytoplasm. The analysis of loss showed a progressive decrease of nuclear TDP43 levels following a small interference RNA. This reduction of nuclear TDP43 was found to alter the TDP43 functionality as assessed by the splicing activity of the protein, and was also found to cause the significant reduction of cell viability as assessed for the MTT test and the caspase-3 assay. The analysis of GOF, which is assessed by inserting into the cells TDP43 inclusion bodies, show the progressive accumulation of TDP43 aggregates in the cytoplasm, which increased with aggregate concentration without any significant modification of the endogenous protein. These cytoplasmic accumulations did not cause any significant alteration of the TDP43 functionality, but was found to be highly toxic, causing a significant reduction of cell viability. We finally quantified the contribution of LOF and GOF by correlating respectively the nuclear and cytoplasmic TDP43 levels to the cytotoxicity that we observed by the MTT reduction test. From the interpolation of the curve fitting, we obtained a contribution of 55% for LOF and 45% for GOF. And similar results were obtained by the correlation of the TDP43 levels to the caspase-3 activation. So these results suggest that both LOF and GOF mechanisms contribute equally to the deleterious effects associated with the TDP43 pathology. Following this data, we took advantage of the innovative super-resolution STED microscopy to study the mechanisms of TDP43 inclusion formation and the involvement of stress granules in this process. We repeated the previous analysis by transfecting the murine motor neuron-like cells with the plasmid expressing human full-length TDP43, but in this case, we monitored the presence of TDP43 by using STED microscopy. The time course analysis that we can see show the progressive increase of nuclear TDP43 nuclear fluorescence, followed by progressive accumulation of the protein into the cytoplasm, together with the loss of the protein from the nucleus. The higher magnifications show better the presence of TDP43 inclusions into the cytoplasm. We also carried out a three-dimensional reconstruction of the whole cell by labeling in red the cell membrane and in green the TDP43 protein. In this way, we can better visualize the nuclear depletion of TDP43, as well as the presence of TDP43 inclusions into the cytoplasm. The quantitative analysis of the fluorescent signals arising from the time course experiment showed that nuclear TDP43 levels significantly increased up to 24 hours after cell transfection, followed by a progressive reduction which appears significant after 48 hours after cell transfection. This pattern is likely to be due to the progressive accumulation of the protein into the cytoplasm, which reached the maximum value 30-40 hours after cell transfections, followed by a progressive reduction of the cytoplasmic levels, probably due to the degradation processes activated by the cells. We next assessed the cell viability of the cells, and we observed that the overexpressed protein caused a significant reduction of cell viability 30 hours after cell transfection. And this cytotoxicity became more intense 40-48 hours after cell transfection, time in which we have observed the the accumulation of the protein into the cytosol together with the loss of the protein from the nucleus. So the toxicity that we observed can result from both loss of function mechanism due to the nuclear depletion of the protein, or from a gain of function mechanism due to the accumulation of the protein to the cytosol, or from both mechanisms. So to dissect the two contribution, we analyze we compare the cytotoxicity that we have observed by the MTT test with the theoretical toxicity that we calculated by the fitting of a mathematical function which considers both the decrease of nuclear defluorescence and the increase of cytoplasmic fluorescence. And from this analysis, we calculated the two coefficients that are respectively associated with the decrease of nuclear fluorescence and the increase of cytoplasmic fluorescence. And this value reported a contribution of about 60% for LOF and 40% for GOF. So also in this case, the toxicity that we observed can result from a combination of both mechanisms that together contribute to the TDP-43 pathology. We also quantified the aggregate sites over time, and we identified 6 classes of aggregates. In particular, we found that the number of large aggregates increased up to 40 hours after cell transfection. And this suggests that the cytoplasmic toxicity that we have observed arises from the accumulation of large TDP-43 inclusions, which, however, were found to decrease at longer time. On the other hand, the small aggregates were found to increase both at early time and a later time. So, considering that at longer time following cell transfection, the small aggregates were found to increase and the large aggregates were found to decrease, we asked whether the autophagy and proteasome systems are activated in our experimental conditions. So by treating the cells with 2 different inhibitors of the proteasome and autophagy systems, we observed that at longer time following cell transfection, TDP-43 inclusions still remain into the cytoplasm of the cells. And this suggests that the proteasome and autophagy systems are involved in the clearance of large TDP-43 inclusions. We next focused our study on the phase separation of TDP-43. Indeed, it has been recently demonstrated that liquid-liquid phase separation with the formation of membraneless organelles is important for normal cellular function, but also play a role in disease, including neurodegenerative disease and ALS. In particular, the phase separation behavior of TDP-43 appears to be correlated with its propensity to form stress granules, which can mature and undergo to liquid-solid phase separation through an irreversible aggregation. To analyze whether TDP-43 protein is recruited to stress granules, we monitored the colocalization of TDP-43 with different markers of stress granules, such as ataxin 2, TIA1, TIAR, EAF3-eta, and FMR1. Our STED images show that TDP43 in green colocalized with all the markers of stress granules in red, as assessed by the Pearson correlation coefficient. In particular, we obtained a strong colocalization with ataxin 2, TIA1, TIAR, and EAF3-eta, and only a minor colocalization with FMR1. Anyway, we also observed that some TDP43 aggregates showed only a minor or no colocalization with stress granules, suggesting that the formation of TDP43 inclusions is driven by different pathways. We also carried out a three-dimensional reconstruction of the whole cell to better visualize the nuclear TDP43 depletion, as well as the presence of TDP43 inclusion colocalizing with ataxin 2, but also the presence of TDP43 inclusion not colocalizing with ataxin 2. Finally, we carried out a time course analysis of the TDP43 cytoplasmic accumulation by monitoring its colocalization with ataxin 2. Our STED images show a progressive colocalization of TDP43 with ataxin 2, which reached the maximum value 40 hours after cell transfection, time in which we have observed the highest cytotoxicity. But also in this case, we observed the presence of some TDP43 aggregates that showed only a minor or no colocalization with ataxin 2, suggesting again that the formation of the inclusions is driven by different pathways, and that the recruitment is not imperative for the formation of the inclusions. So we can conclude that the overexpression of TDP43 in murine motor neuron-like cells caused the deleterious nuclear depletion and cytoplasmic accumulation of inclusions. The toxicity that we observed can result from both a loss of function mechanism and a gain of function mechanism that both together contribute to the TDP43 pathology. In addition, the cytoplasmic toxicity arises from the accumulation of large inclusions, which then decrease at longer time because of the activation of the proteasome autophagy systems. Furthermore, TDP43 appear to be recruited to stress granules formation, but this recruitment seems to be not imperative for the inclusions accumulation. I would like to thank all the research groups involved in this study and you for your attention.
Okay, thank you, Roberta, for your talk. And we have a few questions. So the first is from Sonia Longhi. How was internalization of TDP43 IBS achieved?
The internalization of TDP43 inclusion bodies was assessed by using a specific transfection kit, which is specific for proteins, like the lipofectamine, seems to the lipofectamine.
And then Enas Ortman poses more generic questions. What factors favor LOF and what factors favor GOF?
In our experimental conditions, we observed both mechanisms when we overexpress the protein. So the presence of high amount of protein caused both mechanisms. We stresses the condition to obtain both mechanisms.
Mm-hmm. And another question from Ursula Stokai. Do you see that TDP43 associates with mitochondria in your experimental model? Are inflammatory pathways activated for the loss of function or or gain of function models?
Regarding the mitochondrial localization of TDP43, we did not assess this co-localization, but is an interesting point to focus in the future. And also the second question, I don't remember, sorry Fabrizio.
The second question is, are inflammatory pathways?
Inflammatory pathways, no, we didn't assess the inflammatory pathways, but it could be very important to assess, I think.
Mm-hmm. And there are two questions from Prasad Tamineni. I can select one. Whether large TDP43 aggregates fit into the pocket of the proteasome, how a cell decides when it should send TDP43 aggregates to the proteasome versus autophagy machinery?
We observed that the large aggregates are indirectly, so in our model we assess it indirectly the activation of the autophagy of the proteasome system, but we think that maybe the larger aggregates are degraded by the autophagy system. Mm-hmm. Mainly by the autophagy system.
And then the last quick question from Thomas Meyer. He says, great talk. Did you observe similar effects in other cell types like EC, maybe he means EC293?
No, we observed the same behavior in neuroblastoma cells. No, I did not use the Excel line, okay, but the neuroblastoma cells.
Okay, so thank you very much. I think our time is over, and thank you to the attendees for the many questions. I see that even Nicolas is still busy to respond. He was the first speaker and still busy to respond to the questions. So we have really many of them. So I will ask also Roberta to stay if she can. I know that it's pretty late in Italy, but if you can just stay and type your answers to the remaining questions. And let me thank again all the four speakers for your fantastic talks. Thanks to Nicolas, Jianxing, Witold, and Roberta. In particular, thanks to Jianxing, because he's still answering to questions, and it's 3 a.m. in the morning in Singapore. So it's a sacrifice for him, not just to give a talk, but answer to the various questions. And finally, thanks to Amy Keating for organizing this series of webinars, and to Raluca for her technical assistance. Thank you very much.
Okay, thank you very much. Bye-bye.
Thank you, bye.
Bye, thank you.